My name is Denise Renner, and I am the president and co-founder of GRIN Foundation. I co-lead the organization along with Keith MacArthur. My son has a mutation in the GRIN1 gene that he was diagnosed right before his second birthday, and he's now 8 years old. My son Brett, and this is the session on the GRIN, the GRI gene phenotypes. And what we're looking at here is a result of a survey that we did last year among our patient family population. We had about almost 200 families respond to the survey. And you can see here the prevalence of each individual symptom overall, GRIN. This was before we expanded into our other GRI genes as well. So you'll see here GRIN1 genes, the prevalence of each of the individual symptoms, GRIN2B, GRIN2A, and GRIN2D. And then we moved on to, this is page 24. And this is an accumulation of where families told us they wanted us to focus on the priorities of treatments and cures. And this is the overall most important symptom to treat was intellectual disability and communication. And then you could see amongst the different individual GRI genes, how that might shift within the different GRI genes. Epilepsy being the second most requested symptom to focus on, and mood disorders and behavior and what we now know of as neurostorms as well. So I will turn it, we have 4 panelists on the event today, and Dr. Benke was not able to, Dr. Tim Benke from University of Colorado is not able to join us in person, but he prepared a video for us all to watch. And I will stop sharing here. And Clarissa, if you want to go ahead and load that video.
Hello, my name is Dr. Tim Benke, and I'm a pediatric neurologist at the University of Colorado. And I'm going to talk to you today about GRI disorders, registries for clinical trial readiness, and natural history studies. This is a partnership between myself with Dr. Park and Jennifer Sargent in the University of Colorado and Children's Hospital Colorado, with Steve Tranellis, Hong Chi Yuan, and Scott Myers at Emory University. These are my disclosures. Important ones are funding for this project, which has been through Simon's, GRIN2B Foundation, and CURE, with Dr. Tranellis as the PI. I've been the site PI here, and there are other scientific advisory and clinical trials that I have been involved with, and all remuneration has been to my department. So our hypothesis is that GRI change plus GRI function will result in unique phenotypes and potentially unique treatments. And so the different features that we have been looking to ascertain have been these listed here: developmental milestones, regression, epilepsy, storms, MRI and EEG findings, vision, movement disorders, growth, puberty, medications used for any of these therapies, sleep, behavior, GI issues, autonomic issues, and other illnesses, and longevity. Our approach has been to enroll individuals who have any variation in GRIN, GRIA, GRIK, GRID, and associated proteins. We have had funding initially through GRIN2B, and then now more recently through Simon's Foundation through 2024. And we've also received funding through support at the Children's Hospital Colorado Foundation. This has resulted in key synergies and partnerships, both with Dr. Lemke at Leipzig, Dr. Baines at Columbia, and Simon's Foundation, Dr. Dennis Lau at the Broad Institute, and most importantly through Dr. Tranellis at Emory and the Center for Functional Evaluation of Rare Variants. We have developed what we're calling the GRIN questionnaire, the TGQ. This has been updated to match data collection from what is obtained by Dr. Lemke at Leipzig, and it is now in an online version. This has been updated to reflect parental concerns through interactions with GRIN2B Foundation and GRIN. The consent has been updated to allow de-identified data sharing with our partners. The consent has also been updated to allow recontact for future such studies and updated to allow contact information sharing with patient advisory groups such as GRIN2B and GRIN. It's also been updated to allow annual updates. That IRB has been approved, and because of the changes, those that have been enrolled in the past, we do have to reconsent you. So if you do see a request for reconsent, this is why, is because things have changed since we may have initially enrolled you in the past. We now have papers in process. One of our reviews is now published. The second review, which is a book chapter, we're waiting for the final proofs, and that should be published soon. And then we're looking to publish 2 papers on GRIN1 and GRIN2B in the very near future. So our functional approach has been to perform functional assessments of all GRIN, GRIA, GRIK, and GRID and associated proteins through CFIRV and Emory, and this is led by Drs. Trainellis and Yuan. There's now a revised functional workflow where 8 parameters are assessed to determine net effects of changes at synapses and for non-synaptic receptors. There's now new American College of Medical Genetics, that's ACMG, criteria for determining pathogenicity utilizing these functional changes, and this has been spearheaded by Dr. Lemke. Functional quantitative data now goes into the GRIN portal, allowing correlation with clinical features. This is through work that we're all performing. And the GRIN portal has an algorithm to determine gain of function and loss of function for variants with the complete data set. And this is a feature which is coming soon. And this did require an IRB renewal for this functional work, and that's now been approved at Dr. Trainellis' site at Emory. So this is where we're at. We're pleased that we've continued to make progress. So we've now had a total of 185 contacts, and in some instances, we're awaiting returned consents or mixed and other genetic results that we're awaiting to come back before we can complete the consent. Our goals are analysis of 50 plus with the GRIN questionnaire and functional analysis for GRIN1, GRIN2A, and 2B each. And these will be combined with variants already collected by Dr. Lemke for further analyses. And so here's where we're at. And you can see that we're getting close to our goals with GRIN1. Still have a ways to go with GRIN2A. We've gone past them with GRIN2B, which is great. And here's where we are with all of the others. And again, this is something that's been emerging. And a lot of this has to do with these are very rare. And so it's both that coupled with just getting the word out that people can enroll in this study. So just if you're not aware of it, there is the GRIN portal, and you can see what the address is up here. It's grin-portal.broadinstitute.org. And this is basically your gateway to information on basic information for families, variant analysis, research, and of course, the GRIN registry. This is what it looks like when you go to the family organizations here. And one of the questions you're going to ask is, where is my functional analysis? So the full analysis is provided to your physician at their request, and they can contact me at tim.benke@cuanschutz.edu for this. And it's important to note that gain of function versus loss of function, there are shades of gray. And our view is that this is not clinically actionable, but it is necessary for clinical trial research and natural history studies. Providing this directly to families implies a clinical relationship, and this is a research relationship. And so I think that there could be further discussion on this point, and perhaps this is something we can discuss in the Q&A later on Saturday. Quarterly, the plan is to post these to the GRIN portal as these become available. And importantly, there is information on how to enter the GRIN registry on the GRIN portal, but also here is that key information here. And the main questions: if you haven't enrolled, why haven't you? And if you haven't updated, why haven't you? And it's important to note this is the only pathway to functional analyses to determine gain of function and loss of function. And we believe this is also the pathway through discovering natural history as we collect more information on a yearly basis. Again, this is that web address for that one-stop shop for all. But if you're looking for the unique contact information, this is the contact for Europe, Asia, Africa, or other. And for North, South America, and Australia, it's either Jennifer or Dr. Bain. We're collecting slightly different information, so we hope that you'll register with both. So thanks very much. I look forward to your questions at a later time.
Okay, Dr. Bain, I believe you're up next. And Dr. Bain is from Simons and Columbia University.
All right. Can you see my slides?
Yes.
Great, thank you. Good afternoon. My name is Geoffrey Bain. I'm a child neurologist at Columbia University in New York City, but I've also had the pleasure of working with Simon's Searchlight, for which I'll be presenting their data today. I'm thrilled to see the significant gains as I met the GRIN community just a few years ago, and it's really exciting to see how much has really continued to happen and forging forward in the virtual era during the pandemic. Today, I'll be speaking on behalf of the Simons Foundation project called Simon's Searchlight. I will explain what Searchlight is, how it can benefit families and researchers in the field. And lastly, we'll share some of our data from the GRIN2B, GRIN1, and GRIN2A in order of number of participants. Unfortunately, we only have 2 GRIN2D registrants, and we can discuss a little bit more about why we couldn't include their data today, but we hope to have more of that information soon. So what is the goal of Simon's Searchlight? The mission of this project is to shed light on these conditions, which are genetic disorders causing neurodevelopmental disorders, collected by high quality natural history study data, and really build strong partnerships between different researchers, as you just mentioned with Dr. Benke and several collaborators, as well as promote use of this data for industry and families and family meetings such as today. The first step is really to collect detailed medical and behavioral histories, along with biospecimens such as blood samples from families affected by certain genetic disorders, in this case, the GRIN disorders. What Simon's Searchlight will do will synthesize all this information that's provided and share those results back to the families, such as we're doing today. The goal of Simon's is really to freely share this data and really allow this data as well as biospecimens to be shared with qualified researchers in the field, and to connect participants as well as researchers from around the world in forums such as this. And the overarching goal is to promote a better understanding of genetic changes and hopefully therapeutic interventions in the future. One of the most exciting things that Simon's does allow for is for collection of biospecimens, and individuals who register for Simon's Searchlight have the option to donate blood for research. This can happen remotely using laboratories known as Quest Labs in the United States, but also has happened and continues to happen at in-person family meetings where international participants could also donate their blood. This donated blood is then sent by mail to the biobank at Rutgers University in New Jersey. And the blood is used to make 2 types of research resources. One is a cell line, which we're not going to talk too much about today, but on the next slide, I will talk a little bit about the other resource known as an induced pluripotent stem cell or iPS cell. In my next slide, we'll talk a little bit about this and why they're so important. So what exactly is Simon's Searchlight doing to enable these studies using iPS cells? As mentioned in the very beginning, participants in Simon's Searchlight can donate their blood that's shipped to and stored at the biobank in New Jersey, Rutgers University. The conversion of blood cells to induced pluripotent stem cells happens at the New York Stem Cell Foundation, and the resulting cells are then again stored back at the biobank at Rutgers University. It takes about 6 to 9 months from receiving the actual sample to making these stem cells. And when these stem cells are available, what they can do is be provided back to researchers around the field. All a researcher has to do is provide an agreement with the Simons Foundation to request those samples. We'll review it to ensure that they are a legitimate researcher in the field, and then the samples are shipped to them directly. So who cares about these iPSCs? Well, they're very important cells, because what these cells allow are us to actually go back and differentiate or specialize the cell types. For example, in the case of many of these disorders, we're really interested in the brain cells. We're not as interested in the blood cells. And so what we can do is actually take these blood cells, convert them into stem cells, and then specialize them into the cells that we're interested. Some of the cells we've discussed earlier are nerve cells, microglia cells, astrocyte cells, and other types of cells around the body. And this can really allow for specific cell modeling to be done in the dish, as well as potentially working on drug screening and potentially other drug discovery. So the use of iPSCs can be very important, and Simons Foundation, or Safari, really saves researchers times and money so that they can focus on the experiments and utilize these resources for very nominal costs. But let's get into what this panel is about, which is about the actual information provided by families to the Simons Searchlight Foundation. First, I'm going to start with the GRIN2B Registry in Simons Searchlight. This has been a very active group using the Simons Searchlight platform. And so we'll go through what happens when an individual goes on to Simons Searchlight online, which is step 1 over here on the left. At first, they go online themselves. They don't need a referral. A person can look up their specific registration. The next thing they do is they provide their genetic report, which will be reviewed by genetic counselors and specialists at the Simons Foundation to assure that that individual does in fact have a GRIN2B genetic variant. At that point, they will be given questionnaires that are done electronically as well as with phone interviews to share important medical information. They can fill out many different surveys. They then have the opportunity to provide blood samples if interested. And then the Simons Searchlight platform will allow an individual to provide updates every year about their medical history. And so starting on the left, you can see we've had 116 individuals with GRIN2B sign up, 88 of which have provided genetic reports, which we'll talk about shortly. 66 have completed medical histories. Another 80 have filled out surveys that we'll be presenting today. And 34, which is a great number, have provided blood samples. And we'll talk about some of the specific resources that have already been built by the GRIN2B community. This is a very exciting study, Simons Searchlight, because it's a long-term study. It's been going on for over a decade now, and the goal really is to gather new information every year from individuals. So this is not possible without your participation. The reason why the number of families here is not the same. So we have 28 families who've actually contributed their biospecimens. So 31 individuals with GRIN2B variants. However, 53 family members also provided biospecimens. And from those specimens, there are actually 8 induced pluripotent stem cell lines that are currently available, with 4 additional lines that will be available in the summer. In the next slide, I'll show you which ones they are specifically. There are an additional 8 cell lines that are available, as well as 19 whole blood DNA samples, as well as some saliva DNA as well. This is a list of the various iPSC available resources. So the first 7 you can see are listed up top, and then an additional 4 that are soon coming. 2 of them are from the same variant listed as number 4. So you can see that there's 2 of them. This is a very nominal fee that's being asked for, really just transferring that data and that material over to researchers. It's about $120 per sample to be requested for an individual, and it can be done right online, and researchers can readily sign up and submit those requests now, and we're happy to help you with that process. This slide summarizes the variants observed in the GRIN2B database at this point in time. So we have 80 individuals, and this is as of March 2022. And as you can see, the panel on the left is listing all the different missense variants that are pathogenic or likely pathogenic in the group. There are a handful in the top right of frame shift or nonsense likely pathogenic or pathogenic variants. There's a few with deletions and one additional duplication and one additional splice site variation. All the ones listed here are considered pathogenic or likely pathogenic. There are a few other individuals who may have other variants that are not listed here, and there may be a variety of reasons why they're not included. Their data is collected, but perhaps at this point, their variant is not considered pathogenic or likely pathogenic, so they might not be listed here, but those individuals are continuing to provide information if and when those variants are changed in terms of their pathogenicity. So today, what I'll be presenting are the 52 individuals with pathogenic or likely pathogenic variants who've contributed to that medical history data. And this is through online surveys as well as discussion on telephone interviews. The most recent history interview that we do, we're listing here. So this is looking at the age of individuals who are participants. And as you can see, the majority of individuals that are engaged in the Simon Searchlight group are young children under the age of 20. There are 2 adults who are over 20 years old, but as you can see, the majority of people who are really engaged in the Searchlight community at this point are individuals in those single digits or perhaps double digits range. And it's really going to be important to continue holding on to these groups and really engaging them in the community so we can see what happens as this curve kind of shifts upward and we start to see individuals moving into those teen years as well as adult years. With regard to the developmental and behavioral diagnoses, and these might be the reasons why individuals had genetic testing in the first place, the vast majority, nearly all, have had a history of developmental delay in their younger years. And as older individuals, they have been given the diagnosis of intellectual disability. That's the blue bar on the top. And a very large percentage, up to 3/4 of individuals, also report a language impairment, which we'll talk about shortly as well. About 1/3 of individuals have been given a formal diagnosis of autism, and many other individuals have other behavioral concerns such as ADHD or attention problems, anxiety, and obsessive compulsive disorder. Again, these are neurological disorders or neurodevelopmental disorders. The majority of individuals do have reports of low muscle tone or loose tone or kind of a floppiness or not a weakness per se, but this might have been a diagnosis such as hypotonia given by one of your medical providers. And another 1/3 of individuals also report coordination difficulties or problems with motor planning. There are a number of individuals who have reported having spasticity or tight or high muscle tone, as well as other disorders such as cortical blindness, movement disorders, tic disorders, a small head size or a large head size, and some individuals have been given a diagnosis of cerebral palsy. We're very interested in those individuals who provided information about seizures. And so 52 individuals completed a questionnaire specifically about seizures, and 10 of those individuals, so 19%, or 1 in 5 individuals, reported having had at least one seizure in their lifetime. There didn't seem to be one specific type of seizure. They could be petit mal or absence, also known as staring spells. Simple partial would be one small area of the brain that was involved, as well as other types such as grand mal or generalized convulsions, spasms, complex partial seizures, and tonic seizures. There are many additional medical problems that have been reported by individuals with GRIN2B. Similar to many other neurodevelopmental disorders, 73% have complained of gastrointestinal problems, 58% have eye or vision problems, endocrinologic or growth problems in 44%, musculoskeletal, orthopedic, or joint problems in 21%, GI surgery, such as a G2 for supplemental feeding, as well as a few other disorders such as heart problems, cancer, and kidney and urinary problems are not as high on the list, but certainly are reported in the group. And when we ask on a different questionnaire what medications individuals are taking, just under half of the individuals are taking medications for GI problems. About 23% are taking medications for seizures. And on the right, you can see the types of seizure medications that are being utilized. So in the 10 individuals who have a history of seizures, 2 of them reported taking Keppra as the most effective medication, 1 on valproic acid or Depakote, 1 on phenobarbital, and 1 on medical cannabis. Other medications that have been reported include respiratory or asthma allergy problems, medications for sleep, 20%, problems with attention and hyperactivity, cannabis or cannabidiol, antidepressants, other behavioral medications, sedative medications, and pain medications. One of the more interesting things that we've been recently taking more interest in is looking at what developmental milestones are being achieved by this group of individuals, as well as those with any developmental delay. There's a standardized measure called the Vineland Adaptive Behavior Scale. This is commonly used either in schools or by psychologists or neuropsychologists, and it's also used by the Simon Searchlight group. This graph is what I think of as a developmental growth chart. On the bottom x-axis, you can see the age of the evaluation. On the y-axis is when you use that Vineland Adaptive Scale, what age are they acting, or what age skill set do they have? In this case, 35 individuals reported what they were able to do at these specific ages on the bottom. Each dot represents a different person. For example, the person over here, that individual is an adult who's over 18 years old. However, the person all the way down here was just about probably a few months, because they're just over a year of age. And on the left, you can see what age they would be when they use that specific test. When we plot all the different individuals over time with GRIN2B, what we can see is that individuals do have a delay in their expressive language. However, individuals do gain skills as they are getting older. And so we can't specifically tell you or predict whether your child will or will not gain speech, or to what degree. We can say that on average, there does seem to be growth over time with the acquisition of expressive language. For example, over half of the children above the age of 2 are at a level where they are using some language or learning some language. And after the age of 12, nearly 60% have developed more complex functional language. We can use a similar scale looking at motor development in individuals. For example, here, similarly, individuals do continue to gain skills in their motor development. And after turning 2, over the half of the children at this group are at the level where they may be walking independently. And this is based on 28 individuals who reported this information. So in summary, for the GRIN2B group, we can say that common issues are developmental delay, intellectual problems, language problems, low muscle tone, eye problems, and GI problems. Some of the less common issues, but certainly are still there, are autism, seizures, coordination problems, growth problems, muscle problems, as well as musculoskeletal problems. And so while the GRIN2B did have the most individuals in their group, we are going to go quickly through the other GRIN registry information. So next we'll go through GRIN1, and we can go through a little more quickly since we're a little more familiar with the format. So for GRIN1, 11 individuals have signed up, 10 have provided their genetic report, which I'll show you briefly, 8 have provided their medical history, and 9 have provided some surveys. We don't have any blood samples yet for the GRIN1 group out there, so we hope that we can engage the community with this forum this weekend. These are the variants that are observed in the 9 individuals. These are all pathogenic or likely pathogenic missense changes in the GRIN1 gene. And onto the medical history. This is 8 individuals who provided information. Again, we have no adults in this group. However, we do have a fair number under the age of 5, as well as 2 individuals who are in their teen years. It will be really exciting and important to continue to engage these families to stay involved and stay engaged with providing their information as they get older. Similar to the GRIN2B story, many individuals who have the GRIN1 disorder have developmental delay or a diagnosis of intellectual disability when they're older. About 3/4 do have language impairment. Autism, ADHD, and anxiety are reported in the group as well. Similar to GRIN2B, there is a low muscle tone in most individuals. There's high muscle tone noted in one individual. Coordination problems, movement disorders, as well as cortical blindness are reported in a fair number of individuals in the group as well. Out of the 8 individuals who have reported their information, half of them have reported having seizures. Similar to what I described earlier, there isn't one specific seizure type. They can be grand mal, petite mal, simple partial, or tonic seizures. So seizures seem to be a little bit more prevalent. However, again, we're only looking at 8 individuals. Additional medical concerns that have been brought up by the GRIN1 community include difficulty with growing or gaining weight in many individuals, and similarly about 75% with gastrointestinal problems. Eye and vision problems are noted in about 50% of individuals. There seems to be a little bit more with regards to heart problems, genital problems. One individual has had a G-tube placed for nutrition, and there is one individual who reports having orthopedic or musculoskeletal problems. There are 8 individuals in the group and 7 are taking medication. Most are taking medication for gastrointestinal issues, so 51%. 43% are taking seizure medication. On the right, you can see the 2 medications that have been reported as most effective are Keppra or Perampanel, Fycompa, and levetiracetam. Antidepressants and cannabidiol have also been reported in the group. So in summary, similar to what we talked about earlier, developmental delay and intellectual disability is quite common, as well as language impairment, tone problems, coordination and movement problems. Seizures do seem to be a little bit more of a problem in this group. Difficulty growing and gaining weight, vision problems, GI problems. Some of the less common issues that are reported, however, are still being noted in the group include cortical blindness, heart problems, and genital problems. And we'll finish up with the GRIN2A registry and Simon Searchlight. So we do have 15 individuals who have signed up online. 9 have provided their genetic report. 5 have shared their medical history. 9 have completed surveys. And we do have 1 individual who has provided a blood sample. And again, we do encourage everybody to consider enrolling and providing information on an annual basis. In the GRIN2A group, there are 9 individuals. The majority of individuals also have pathogenic and likely pathogenic missense variants. However, there is 1 individual who has a pathogenic splice site variant listed here. There are 2 other variants of unclear significance that have not been shown, and these are all of the collected data from March, so just last month. In brief, I'd like to say thank you very much. Unfortunately, we don't have much more information from the group at this point, but we really want to thank the community for being engaged and using Simon Searchlight to continue gathering information to share back with you and to share with other researchers. I'm happy to answer any other questions. And I really want to thank you because this wouldn't have been possible without your willingness to participate in the Simon Searchlight community. Thank you and have a nice day.
Thank you, Dr. Bain. I know we have a couple of questions in the chat, and I know we have some time allocated towards the end, but I just wondered if there's something specific to your presentation. I know the question has been asked with these various registries. Sometimes these questions can be technical. Is there somebody at Simon's that can help with the answering some of these more technical questions?
So what I'll say from Simon's is that families do sign up online, but they do get to speak with humans as well. So there are specific coordinators, and I think Curtis Weaver is in the chat right now. He's one of the research coordinators. They will work with individuals on a one-by-one basis to make sure that all their questions are answered, that they're requesting the information that we need, and that we're giving you the information that you need. Certainly it is a process. So for Dr. Benke's study with Dr. Trinellis. Jennifer Sargent will sit down and go through the project with you step by step. There is what we call a consenting process, meaning that to be an ethically sound study anywhere around the world, an individual will sit down with a family and tell them the risks, the benefits, and the entire process to really be transparent about the process, what you're being asked to provide. And that's an opportunity before you sign on any line to ask any questions, to provide any feedback so that you really are clear about what information you're being asked to provide, what you're getting yourself into, since this is very personal information, and really what will happen with that data and that information over time. So I encourage families to really even just ask, because even just asking might answer some questions, provide some relief. I think parents are not always sure what they want to sign their own kids up for. And a lot of this is just data information and collecting information from parents. And we really need to know the good, the bad, and the ugly to get a good understanding for what's going on and what's affecting families. Again, an email or a phone call does not imply that all of your information will be shared. There's a full consenting process where you have the opportunity to talk through what information you want to share, what information you might not want to share. Just because you sign up does not mean you have to donate blood, go through these processes, etc. Really, I think any information families are willing to provide can be helpful for the big picture. Hope that answers your question.
Yes, thank you. Thank you so much. And just one more question, and maybe this can be answered here, but from a parent's perspective, if parents have provided blood samples to other universities, it certainly would be beneficial to be able to share that with other organizations such as Simon's.
So I'll go the other direction. So Simon's basically, one family once said to me, Simon's is almost like the library. Simon's will collect all this information, will collect these biospecimens, and they freely will share that with other providers. At this point, and I think my Simon's colleagues in the background can chime in if I'm wrong, I don't think we can collect information from other locations, unfortunately, because it is a specific preservation process. But they are very happy to work with individuals and answer any questions they might have. I think that's one of the hardest things that families and research teams and patient advocacy groups are faced with, because they want to move the field forward, but you also don't want to give blood samples out all over the place. We're all children, and we want to take care of everybody and really try to consolidate as much as we can. Dr. Benke had mentioned that Dr. Benke, Dr. Trinellis, Dr. Lemke, and myself are really trying to work together so that all of this information can really be housed together, so we're not duplicating efforts to give everybody parent survey burnout.
Yeah, and blood draw burnout. Thank you so much. Okay, so we're going to move on with Dr. Allan Bayat from Philadelphia Epilepsy Hospital in Denmark.
Hi, 1 second. So, thank you very much for inviting me. My name is Allan. I have a medical background. I'm a pediatric neurologist in Denmark, and working at an epilepsy hospital, but we are primarily interested in genetic epilepsies, but in general, neurodevelopmental disorders. I've been working on my PhD for the past 3 years, and now it's done. And I have focused my interest on GRIA genes and GRIA-related disorders. And we are actually doing exactly the same as Tim Benke and Trinellis, but I'm collaborating closely with Johannes Lemke. So I will give you an update about overall the GRIA phenotypes that we know of. I'll give you an insight about the Leipzig registry. Tim made it easy for me, so it'll be more or less what he said. But down below, you have my email contact. And if any GRIA families out there are interested with help to go through the registry, I would be willing to help them with some of the technicalities if they have problems. So let's begin. So I wanted to give you an insight about GRIA to just understand the background. So in the brain, we have two very important types of cells, two different neurons. The yellow one are what we call the excitatory neurons. These are the ones that kind of generate electricity and activating cells around them. Then we have the inhibitory neurons that are kind of shutting down the excitatory neurons. So it's very simple that there is constant a balance between excitation and inhibition. And if a mutation occurs in a patient with GRIN or GRIA related disorder, then it'll shift this balance between inhibition and excitation, for example, in another direction. So you might have too much inhibition or you might have too much excitation, and that causes neurodevelopmental disorders. So what we're interested in today is to talk about the GRIA genes that encode the AMPA receptors, and the AMPA receptors are located here on the cell surface of these excitatory neurons. They're located just beside the NMDA receptors that are encoded by the GRIN genes. So this is what an AMPA receptor looks like and what it does. So it's located here in the cell surface, and you can see that once it closed, it doesn't really create any current. But when it opens, the current goes up, and then when it closes, the current goes down. And what happens is, if you have a mutation that that destroys the channel, then it'll be the current will be either completely flat, there will be no current, or it'll be diminished. So this is what we call loss of function mutations. But we can also have mutations that have the opposite effect, where the channel stays open too long. So you have a gain of function, the current is too high, or it doesn't really go down. So that's how the channel works. This is another figure of the channel just to show you that it's made out of different subunits. So it's currently made out of 4 different subunits. And you can see that these are identical pairs. So you have 2 subunits here that, for example, are often encoded by the GRIA2 or GRIA3 genes. And then you have another pair of subunits encoded by one of the other genes. So there are 4 genes, as you know. And they encode these subunits that go together and create the AMPA receptor. So what do we know based on the available literature that has already been published? Well, we know that all patients have a global developmental delay that is apparent very, very early in life. So it starts in infancy or in early childhood. But there is a range in symptoms. So some can be very mildly affected, while others may be profoundly affected, more severely affected. So I'm highlighting the mild spectrum and the severe. And then, of course, there'll be a lot of patients that will be in the middle. So in the mild spectrum, we have boys and girls that develop completely normal for the first 3 to 6 months. They reach their developmental milestones, and parents have no idea, and neither do clinicians, that there's anything wrong with them. And then about 3 to 6 months of age, they start either they regress, so they stop developing further, or they may even lose some of the milestones that they have learned. And what we know is the problems that they face are, for example, motor coordination problems with social interaction, language abilities early in childhood. The mildest patients learn to walk, they'll even learn to run, but may have an unsteady gait. Some don't have epilepsy and they never develop epilepsy. Those that have epilepsy are easily treated with medication. And the epilepsy seems to start very early in life, so around 6 to 12 months of life. So that's in the mildest spectrum that we know of. In the severe spectrum, patients, the children, the infants are affected just after birth, just the first day, the first hours of birth. you can see that they're either very, very flaccid, so they're hypotonic with a reduced body tone, or they can be very, very stiff. They might have feeding difficulties because of these tone problems. There might be very limited or no eye contact. And eventually, some of these patients in the most severe end of the spectrum may never learn how to walk or speak or interact, which is really sad. And they often tend to have early onset epilepsies that are really difficult to treat, despite treating them with several different kinds of medication. So that's a spectrum. And then there's those that are in the mild or in the severe, and those that are in the middle. There's also additional symptoms, and I'm just mentioning on a few of them. There are a lot of behavioral problems, autistic features. So not all have a formal autism diagnosis, but they have features that seems to be these limited social interactions, repetitive behaviors. Some may have be very hyperactive. They can have reduced attention spans. They may have short temper, be aggressive either towards themselves or their surroundings, be anxious, or be very sensitive towards external stimuli. So loud noises, too much noise, too many inputs can really affect them. They may have stereotypies, which, for example, can be head flapping of the hands, rocking of the head, or rocking of the body. There are often speech impairment. The majority have no or very limited meaningful speech. Some can communicate with short sentences or short or simple words. As I said, there are problems with both gross and fine motor skills. So some patients have hypotonia, so reduced body tone, while others are very stiff, are very irritable, and they may later progress to have spasticity or cerebral palsy. And these patients, all of them have this wide, unsteady gait. Some have completely purposeful use of hands, but have fine motor problems. For example, buttoning your shirt or drawing, while others have had purposeful use of hands, but lost it early in life. And then others may never have reached a normal use of hands. So they might not even be able to hold or grasp something on purpose. Then there's a lot of patients that do seem to have different sleep issues. So sleep can be normal, but in some, they can have problems falling asleep or staying asleep, which is really a problem for some. We know of patients that can stay awake for more than 1.5 days, which is really devastating for both caregivers and the patients themselves. So some of the important questions is, we know a lot about the phenotypical spectrum of GRIA-related disorders, but there's still really much to learn. So how do we better understand the phenotypical spectrum? And how do we understand why some mutations give some symptoms while others give other symptoms? So why are some patients only mildly affected? Why are others more severely affected? So why is there this genotype-phenotype correlation? And can we identify any clinical biomarkers? So we might have a family with a son or a daughter who has a mutation in one of the GRIA genes, and what we want to know is this gene likely to give a loss of function in the AMPA receptor, or is it going to give a gain of function? So our lab and Steve Tronellis's lab will be able to solve this question, but not all people know of us, and some may just not have access to us. So what we actually want to find out is, what if a patient has very early stiffness, for example, early in life? Could that be a sign of overactivity in the GRIA receptor? And could could hypertonia or being flaccid, for example, be a sign of a loss of function. So these are some of the clinical biomarkers that we are trying to identify in order to counsel the families and other clinicians to try to find proper treatments. So in order to reach some of these, to answer some of these questions, of course, we need to have a database. And as you heard, there is the US database that is actually pretty much the same questions as in the Leipzig database, but that you saw, they have collected an amazing number of patients with GRIN disorders. Our group in Diana-Lund, in Danish Epilepsy Center, have worked with Lemke's group for the past 5 to 10 years. And during the last year, we have joined forces, and now the GRIN database is also a GRIA database. We haven't started to formally collaborate with Tim Benke and Steve Trenelis, but surely this is something we need to do as well for GRIA related disorders. So you have already seen this slide. So this is an easy way to get to the GRIN registry. I'll jump over this. There's also this link up here that will immediately get you to the registry, but why choose that when you can go through the GRIN portal? So once you go to the GRIN portal, if you haven't been there yet, you will get some questions on the first pages. So you need to, of course, give the name of your child, your own name, your own contact information, for example, email. Then you need to agree to share clinical data with us by saying yes and yes and yes. So you declare that you're then on the numbers. So these data are, of course, it might be the referring clinician. So we really like to know whether it's the clinician or it's the family that are giving the data. So you'll be asked for this. And then over here on the right, when you start typing in about the genetic information, of course, you can choose which of these genes, either the GRIN or the GRIK or the GRIA, which of these genes are relevant for your child. And then if, so filling out this database takes time. It might take a few hours, so you might get tired and want to do it over a few days. So you can always save and get back to it here by clicking down here, and then be aware that you will get a return key and a return link that you need to access the database again. And if you lose it, just contact me or contact me. Feel free to contact me, and I can help you to get the link back. So these are some of the questions that we will ask. So there will be questions about the family history, birth, pregnancy, the early developmental milestones. There will be additional symptoms that we're asking for, comorbidities, whether there has been any epileptic seizures, and if so, what kind of seizures. If you have EEG reports, you can upload the reports so we can have a look at it for you. Any MRIs, brain MRIs that have been done. And again, additional questions about, for example, problems with gastrointestinal tract, puberty, etc. You've already heard a little bit about this. So currently, we are very lucky to have 30 patients with GRIA-related disorders in the database. It's mostly children, but we do have a handful of adults. So this figure is just to show what it is that I'm trying to do here during the next 3 years for my research. I will start, of course, with the data that we have collected from the GRIA families. Then in our lab, I will try to take the human variants that we have. If we don't know whether the variants are actually affecting the AMPA receptor, I will try to look at it in a cell module. I'll try to do some patch clamming and try to determine down here whether the specific mutation is affecting the AMPA receptor, and if it's giving a gain of function or loss of function. And we'll try to see if there are any medications available that can reverse these gain of functions, for example, block the receptor if it's too active, or open with the receptor if it's closed, and then eventually get back to you or your clinicians to tell you the results of these, and hopefully help you to find precision medicine if it's available, or if it comes on the market for future studies. So that's basically it. And once again, below are my contact information. Feel free to contact me if we haven't already been in contact, especially if you're a caregiver family for patients with GRIA related disorders. That's it.
Thank you, Dr. Bayat. I know that we do have links for the registration on the GRIN website, but we'll be adding the GRIN portal there as well, so folks can find it more easily. We did have a general question in the chat that I think probably you can answer. One that we get a lot is that if we share a variant with another patient, that particular variant, and that particular variant is loss of function or gain of function, will that variant always be loss of function, gain of function, or are there sometimes changes within the functional analysis?
I would say it's pretty fixed. So if we find that it is a loss of function, so it's two things to say about this. So there are different technical tests that you can do to express these AMPA receptors. And if you only look at one subunit, you might find on a few variants that it is a loss of function. And then when you assemble the subunits, you might find out that it is actually something else. But sorry, my little boy is coming to say hello. Say hi. Yeah, so the short answer is yes, it's fixed. So if something is a loss of function, then it is a loss of function. And if it's a gain of function, then it is a gain of function. But of course, the lab work needs to be done correctly and properly. But I guess those labs that are involved are doing a tremendous good work. So long answer, sorry.
Great, thank you so much. And now for our last speaker, we have Dr. Geoffrey Swanson from Northwestern University.
Okay, thanks for inviting me to talk about our work on GRIK genes. You probably haven't heard too much about those because so far there are the fewest number of patients that have been identified with variants in this particular family of glutamate receptors. So I've titled this GRIK phenotype, GRIK2 and beyond, just to make the point that GRIK2 is not the only kainate receptor gene that variants exist in patients. But I also want to point out that there is really no GRIK phenotype as in the singular sense. There's a variety of phenotypes, even among patients or individuals with the same variant. So we shouldn't think of or search for a single defining phenotype. So the way I personally entered into this field was through this young lady now several years ago. At the time, she was quite young, and she was diagnosed with Angelmans-like phenotype, global developmental delay, like it's seen in many patients, and hypotonia, characteristically very social and smiling often, and now she is much older and she has developmental delay, intellectual disability, an ataxic gait and chorioathetoid movements, so involuntary movements and postural abnormalities. But one thing I also want to point out is that her MRI is normal. She doesn't have any kind of structural changes at the level of the MRI. So whole exome sequencing found a GRIK2 variant that changed the protein encoding residue 657 from an alanine to a threonine. I should warn you in advance that I'm going to talk about phenotypes both in the humans, but also to a small extent in the mouse model that we've made for this the variant that this young lady has. as Allan showed, this is what a receptor looks like. In this case, this is a kainate receptor. And because kainate receptors are probably not familiar to most people, I want to point out that they're the GRIK1 to 5 genes. They are responsible for a variety of functions in the CNS that are not as well characterized as AMPA and NMDA receptors, but include balancing excitation and inhibition, fine-tuning neuronal excitability, and other functions that I've shown here. The other thing I want to point out is all the reported variants thus far change protein structure in this little boxed domain. That's an important domain for communicating between where glutamate binds in this LBD or ligand binding domain and where the channel pore opens here in the membrane. But there are other variants outside of that specific domain that we haven't reported yet. So this is the, right now, the comprehensive list that I know of. You can see there are variants in GRIK2 and GRIK3 and GRIK5 that are damaging. I apologize for all the abbreviations, but global developmental delay is GDD and autism spectrum disorder is ASD. The purple box shows the variants that we reported recently. And also it shows that there are a number of individuals that we've collected information from with the same variant. So this is alanine 657 that the first patient had, and then a couple others with conserved variants, conserved mutations. But again, there are other variants in GRIK3 and GRIK5. Interestingly, in GRIK5, nearly all of the patients with variants in that gene have autism spectrum disorder that we've identified. The other specific variant that I want to point out is one that was just communicated to us from the clinicians, and that is this loss of function terminating variant, which would be the first such in the GRIK2 gene, and that person has autism. Okay, so I told you in this box, this is where the variants occur that we've described, and they have two general kinds of effects on receptor function. The first one is this isoleucine 668 variant, and that is thought to disconnect the energetics of binding in this LBD area to channel gating. So it is a receptor, it gets made, it gets put on the plasma membrane, it just doesn't work very well as a channel. And then there are a couple variants that destabilize the closed conformation. So that means that the channel will just tend to pop open very easily, even when there's only perhaps one glutamate bound to one of these 4 subunits. And so I don't want to belabor the currents here that you can see. These are currents from HEK cells transfected with different mutant receptors that this is a way in which we characterize how the function of these channels change, and it's very similar to what has been described previously as trying to understand whether they're gain of function or loss of function mutations in these genes. And the point I want to make here is that it's actually quite difficult to determine that, categorize them either as gain of function or loss of function in many cases. And I'll tell you what it is. So here on the left side is what we call the wild type GluK2 receptor. And we're putting on glutamate in the gray bar for a very short time or a longer time. That's to measure different parameters about channel function. And here are the 4 variants that we described recently. So what I think you can hopefully appreciate is that if we look at specifically this A657T, which is alanine to threonine change that was originally described in Katie, the young lady I showed you before, we would call that a gain of function because there's more current passing through the receptor during a specific amount of time, and it's more sensitive to glutamate. And so I boxed on the right side here in our little table, you can see that deactivation, one of these parameters we measured, goes from 2.5 milliseconds up to 212, so about a 10-fold or 100-fold change. It's slower. And a similar sort of slowing of a different parameter called desensitization. So in most ways, we would characterize that as a gain of function mutation. And then on the other side, we have this loss of function mutation. And it's a loss of function because the channel gating is faster, so it stays open a shorter period of time, and it lets less current, lets ions through the channel. And there's much less of this receptor expressed on the plasma membrane as well. So there are fewer receptors and less current going through the receptor. So that's a pretty easy way to categorize this channel as a loss of function. But then there are these receptors that are somewhere in the middle. They have features of both gain of function and loss of function. And that would be these 660 variants. So they get very much slower, so that you can see deactivation again goes from 2.5 by 100-fold slower to 160. But at the same time, there are many fewer receptors of these of this type made that make it to the plasma membrane to be functional. So they're slower, but there are fewer. So what that means in terms of gain of function, loss of function is not really obvious. And the other thing to keep in mind is that these are analyses of defined receptor combinations. So what I just showed you was a comparison of all GLUK2 receptors. We would call this a homomeric GLUK2 receptor. And then some of the GLUK2 mutant receptors or variants. Again, all of the same subtype. So these are both homomeric receptors. And the differences between the receptor parameters are going to be greatest in this type of comparison. But if we think about what that means in a human, in a mouse, and any kind of organism where these receptors exist in their native form, in fact, it's not as simple as that. So we have other, in fact, de novo variants occur on one allele of two. So you get a mixture of the wild type and the mutant allele when these receptors are assembled. You also have other subunit proteins that complex with the receptor. So in the case of kainate receptors, you might get a GLUK5 subunit. So that's in blue. And now your variant protein is really only one of the 4 proteins that make up this receptor. And then it gets even a little more complex because they have proteins called auxiliary proteins. In the case of kainate receptors, that would be something called NETO2, which also bind and change channel function. So when you get to something that is approaching a receptor that looks like a normal neuronal receptor, it gets pretty tough to understand whether this is really a gain of function, a loss of function, or somewhere in between that's tough to predict. Doing this sort of analysis, as I think you can appreciate from the other talks, it does inform predictions about what might be happening in patients and also in other organisms. So I want to just touch upon the phenotypes of two individuals with 2 variants, the alanine threonine variant I mentioned previously. And the idea here is to try and understand whether there is a genotype-phenotype correlation. That is, do individuals with the same variant show the same behavioral, developmental phenotype? And I'll give you the answers to the first approximation, they do. And so in the case of individuals with this variant, they all have initial diagnoses of hypotonia or developmental delay. They share intellectual disability. And I think most notably, they also share this delayed motor development that sort of plateaus or stabilizes in a ataxic or unbalanced gait. So they have a wide range of balance related issues. And similarly, along with some other aspects of their disorder. There are some features or phenotypes that are found in one or a set of the 7 individuals. And if we compare those individuals to the folks with the T660 or the threonine 660 variant, you can see that these also are fairly conserved in terms of their phenotype, but they're very, very different from the kids with the 657 variant. So they have initial diagnoses, often of seizures, and in the case of these individuals, the lysine variant seems to be much more severe than the arginine variant. So they all have intellectual disability. The folks with the lysine variant are not ambulatory, or they have limited degree of walking. The kids with the lysine variant have epilepsy, quite serious epilepsy that's often refractory in most cases, and then cortical visual impairments. So, and then perhaps the thing that really distinguishes them from the other group of kids is that they do have structural abnormalities in their MRIs. And that is sort of surprising, because these channels, you probably can appreciate from my quick overview of the currents, these look very similar to the alanine 657 threonine variants in terms of their function in a dish when we record their currents. We had the opportunity to look at a series of MRIs taken from one of the kids. And what I think you can hopefully appreciate is that you have these structural abnormalities that include thinning of the corpus callosum. You have hyperintensities and increased volume in the extraaxial spaces. And then you see some initial signs of atrophy in the hippocampus, and it's not as obvious here. The hippocampus is this little structure here. Cerebellum also shows some initial signs of atrophy, although it's not as obvious in these MRIs.
Dr. Swanson, I just wanted to let you know we have 2 minutes left. And
Okay.
I apologize.
Okay, well then I'll pretty much wrap it up, because I'm about halfway through. So okay, so why don't I just stop here then and say that missense mutations in kainate receptor genes can be causative for neurodevelopmental disorders, and the variants that alter their function in a similar way, like I said, in a dish, can cause very different phenotypes in the children. And that includes one that possibly causes a degenerative phenotype. And at least in the small population of individuals, there does seem to be a genotype-phenotype relationship. And I will skip over the mouse stuff.